Hello and good evening, everyone. Um, Welcome to the week 6 live class session of Fundamentals of Protein Chemistry. Yeah. I am Srijan Sen from IIT Kharagpur, and today I will be taking your week 6 live class session. Okay, and I'm sharing my slides with you all. I hope my screen is visible to you all. If that is so, so let us talk about the week 6 live class session, where mostly we will be talking about enzymes and their classification and the mechanisms of the different types of enzymes. So what are basically enzymes? So let me introduce a bit to you all about enzymes. Like enzymes are proteins. Just a minute. Yeah, sorry for the delay. Yeah. Like I was telling, enzymes are proteins. It's similar to proteins. It's just like they have a different role to play, or a very important role to play. So it increases the rate of the reaction by lowering the energy of activation. That is, they catalyze nearly all the chemical reactions taking place in the cell of the body. And the beauty about enzymes is that they are not altered or consumed during the reaction, and enzymes are reusable. So enzymes are known as biological catalysts, like we have the chemical catalysts. So enzymes are also like biological catalysts. So they have shared properties with chemical properties, among which it's like that, like the catalysts are neither consumed or produced during any the course of a reaction. So similarly, it's that enzymes are also neither consumed or nor produced. Similar to catalysts, enzymes do not cause reactions to take place, but they basically enhance the rate of the reaction. That is by lowering the activation energy barrier, and they alter the rate not by equilibrium constants of the reaction that they catalyze. However, there are a few differences between the enzymes and chemical catalysts. So, enzymes are proteins. Enzymes are highly specific and produce only the expected products from the given reactants or substrates. Enzymes may show a high specificity towards one substrate or exhibit a broad specificity using more than one substrate. And enzymes usually function with a moderate pH and temperature range because these are proteins. So at extremes of pH, temperature, or difference in the ionic strength and all, it will just be of no use. So enzymes will just be non-functional. So it requires some proper an ideal pH and temperature for doing it. So what does the enzymes do? So like a normal reaction, the energy barrier that you see over here made of rocks is basically very high. Okay, so it is very difficult. So you have say 4 to 5 frogs on the reactant side, and this is the product side. So the number of frogs that would be able to cross this barrier and reach the product side is relatively less, because there are no because the energy barrier being relatively high. So the reaction proceeds at a slow rate due to prohibitive activation energy threshold. On the other hand, if it's an enzyme catalyzed reaction, so if you assume an enzyme to be a person or someone, and they remove these rocks for the frogs to cross this from the reactant to the product side, so that is what is done by enzymes. They lower the energy barrier, and they cross from the reactants to the product side. So the reaction proceeds at a significantly faster rate as the energy activation energy threshold is reduced. So what you can understand is that enzymes do not directly take part in the reaction, rather they help you in crossing the barrier from the reactants to the product side by lowering the energy barrier or the activation energy. So here comes the concept of free energy, where more free energy, that means less stable, greater work capacity. Now what enzymes do is they help the reaction to proceed in the forward direction, that is by lowering the free energy, that is delta G less than 0, which is equal to like the lower the delta G value, that will be higher will be the feasibility of the reaction to occur. So more stable and less work capacity is being done to reach from the reactant to the product side. So when the free energy is greater than that of the reactant is greater than that of the product, the free energy changes for the reaction is negative, so the reaction becomes more feasible and spontaneously the reaction occurs. Now when the free energy of the product is greater than the reactant, the free energy change of the reaction is positive, so the reaction takes time to proceed, or the rate is slower, and the chances of the reaction to occur is relatively less. However, from if you have the product and there is a dissociation or something from the product to the reactants, that is a much more feasible reaction. So enzymes, although they are proteins, but they have different components. Like you have the apoenzyme, that's the protein portion, but they are inactive because they need some supporting elements, which we call as the coenzyme, which are usually your cofactors, different prosthetic groups that comes and attaches to the particular protein. And when they attach to this protein, it incorporates the substrate and carries out the reaction. So the components of an enzyme includes an apoenzyme, that's the inactive protein portion. You have the cofactors, that's the non-protein portion, or in other words, you can call it as the activator of the apoenzyme. The entire of coenzyme and apoenzyme forms the holoenzyme or the whole enzyme that is in its active form, that recruits the substrate to carry out a particular reaction to get into the product. So cofactors can be of two types. It can be your ions or coenzymes. In ions, you have the activator ions that are loosely bound, and upon carrying out the reaction, they simply dissociate into the solvent. And the other ones includes the metal ions or the metalloions. So metal protein interaction is very strong, and those are tightly bound to the protein and keeps it activated. So that is one category of cofactors. The other is coenzymes, that includes your co-substrates that are loosely bound, and different prosthetic groups that are tightly bound to the apoenzyme. The entire story is like this cofactors binds to the apoenzyme to form the active holoenzyme that carries out the reaction. So an enzyme also has different portions. So if you have a protein, or an enzyme protein, you might be knowing that there are several catalytic sites where the reaction occurs. You have the binding site where the substrate binds in place. It can be possible that the catalytic site and the binding site are same or maybe different. So probably you have a catalytic site at some different place and the binding site is somewhere different. That is also perfectly okay. And even it is also like that, wherever there is the catalytic site, there is the place where the substrate goes and binds. So the area on the enzyme where the substrate or the substrate attached to is called the active site of the particular enzyme. So you see over here, you have a substrate entering the active site of an enzyme. You have a green colored substrate, but the active site is not well made up to take up that substrate. So the enzyme changes its shape slightly as the substrate goes and binds, and then you have the enzyme substrate complex followed by the reaction leading to the enzyme product complex, and the products dissociate, leaving the active site of the enzyme, and you get the products. Now, when we are talking about binding, there are two models that explains enzymatic binding. The first one is the lock and key model, the other one is the induced fit model. So what does these two models indicate? It indicates the theory of enzyme kinetics. So what happens in the lock and key models? From the name you can understand. So you have a lock, you have a key. When the lock does not fit the key, it cannot open the door. If the key cannot fit into the lock, the door cannot be opened. So the opening of the door is equivalent to the reaction occurring from the reactants to the product. So lock and key from the model, if you can understand that the active site of the enzyme should be suitable enough to take up that substrate. So if you have a V-shaped active site, it can take up only substrates that fit into that particular shape. Okay, so otherwise there is no possibility of the enzyme to go and bind. So what does it mean that the active site has a very rigid shape? It could be because of the rigidity of the enzyme or due to the rigidity of the entire catalytic site. So only substrates that matches the shape of that particular enzyme or that particular site can only go and fit over there. The substrate is the key that fits the lock of the active site. So it is an older model, however it does not work for all the enzymes. But when this model was created, one bigger advantage of this particular model is that it is very specific. Like if you know that a particular DNA or a particular peptide small molecule should bind that particular binding site, they are highly very specific because it cannot go and bind to random peptides or other ligands, but it has a perfect specificity for that particular ligand, and only then it will bind. The other model is the induced fit model, where the protein or the enzyme has the ability to change its shape, or the catalytic site gets uh, modified to a certain extent. Like the active site is more flexible, not that rigid, and the shape of the enzyme, the active site, and the substrate adjust to maximize the fit, which improves the catalysis. That is a bigger advantage of this particular model. And this model works very well with a wider range of enzymes, because a lot of enzymes can be able to take up the substrate if it works in the induced fit model. But however, at the end of the day, what we want is the substrate to get converted into the product of our interest. In whichever model it works, it's absolutely fine. But why is it important is because about when we speak about the specificity, it's like the lock and key model works well. But if we talk about the catalytic ability or the catalytic efficiency, the induced fit model is a perfect fit. Now, this is about the enzyme nomenclature, where there are so many ways of talking about enzymes. You don't need to remember each of them. If you want, that's absolutely fine. So it is actually indicated by a four-digit number code, where the first digit is the major class. You have the second digit as the subclass, the third digit as the subclass, and the final digit is the of the specific enzyme. So like 2.7.1.1 is the glucose phosphotransferase. So 2 is the class name, that is transferase. 7 is the subclass name, phosphotransferase. 1 is the subclass hydroxyl group as an acceptor, and specific enzyme, that is D-glucose as phosphoryl group acceptor. So the enzymes can be basically classified as different types. There are 7 different classifications or categories, including the oxidoreductases that carries out the redox reactions. You have transferases, hydrolases, lysases, isomerases, ligases, and the translocases. So each of them have different functions, carrying out the redox activity or ligating the ligating different things, like you have the ligases. If you want to change the um, stoichiometry, you or the isomeric merism, you want to undergo isomerism, you go with the isomerases. You want to remove or add water, you have the hydrolases. It's And then if you want to go for translocation, you have the translocases. So there are different enzymes with different activities. What you need to know over here is about the catalytic efficiency. So most enzyme catalytic reactions are highly efficient, proceeding from 10 to the power 3 to 10 to the power 8 times faster than uncatalyzed reactions. Typically, each enzyme molecule is capable of transforming 100 to 1000 substrate molecules into product each second. The number of molecules of substrate converted to product per enzyme molecule per second is called the turnover number. So you have the enzyme classification into 6 classes. You have the oxidoreductase, transfer of reducing equivalents from one redox system to another, like the alcohol dehydrogenase, lactate dehydrogenase, cytochrome oxidase. Then you have the transferase. The functional group is transferred from one compound to another, like you have the kinases, transaminase, phosphorylase. The third one is the hydrolases enzyme that cleaves the CO, CN, CS, CO, etc. bonds by adding water across the bonds and forming new oxygen bonds. Like you have the acid phosphatase, that's important in the digestive process. You have the lipase enzymes. The fourth one includes your lysis, so that cleaves the COC and CS bonds. That is also, but not so, without the addition of water and without any oxidizing or reducing substrate. So it's very similar to the previous one, that's hydrolase, but here you're not adding any water or chemicals for doing it. So you have the aldolase, fumarase, carbonic anhydrase. We know about isomerase, so it catalyzes intramolecular rearrangements of functional groups that reversibly interconvert to optical or geometric isomers, the triose isomerase, mutase, phosphohexose isomerase. Every one of you have heard about ligase, because anyone who does cloning, recombinant DNA technology, you will must hear about ligase. It's for ligating or joining of the two uh, sticky ends. So it catalyzes the biosynthetic reaction that form a covalent bond between two substrate utilizing ATP ADP interconversion. You have the DNA ligase for used in rigorously in recombinant DNA technology for ligating of a vector and insert with sticky ends, complementary end sticky ends, and the other one is the glutamine synthetase. Um, so when we speak of biocatalysts or enzymes, we have to know that there are certain environmental factors that play an active role in affecting the catalytic efficiency of the enzyme. So there is an optimum temperature, the temperature at which enzymatic reaction occurs fastest. So if you titrate, so this is a titration reaction where you check the enzymatic reaction at different temperatures. So it is also dependent on the activity of the enzyme at a particular temperature. So for example, when you set up restriction digestion or ligation reaction, there is an optimum temperature of 37. So 37, or the body temperature, is the one where is the temperature that you basically use for where you see the uh, optimum activity of the particular enzyme. Now pH also affects the rate of the enzyme substrate complexes. Some enzymes are optimal at 7, some prefer acidic or basic conditions. You have pepsin that works in pH of 2, and there are certain other like amylase, the human amylase, that works at 7. There are some xylinases. You have trypsin that works at alkaline pH. So it is not only about the enzyme efficiency, but also about the amount of substrate that you are adding. So the rate of a reaction increases as the substrate concentration increases, as long as it does not reach a saturation and there is not enough enzyme left to bind to the substrate and carry out the reaction. So the maximum velocity Vm, all the enzyme is combined with the substrate. So here the enzyme activity increases and it reaches a stability. The maximum activity occurs when the enzyme is saturated, when the enzymes are in the binding site. So the catalytic power is defined as the ratio of the enzyme catalyzed rate of a reaction to the uncatalyzed rate. For example, you have the urease. So at 20 degrees, the rate constant of the enzyme catalyzed reaction is 3 into 10 to the power 4 per second. The rate constant for the uncatalyzed hydrolysis of urea is 3 into 10 to the power 10 per second. And 10 to the power 14 is the ratio of the catalyzed rate to the uncatalyzed rate of the reaction. So specificity is defined as the selectivity of enzymes for the reactants upon which they act in an enzyme catalyzed reaction. None of the substrates is diverted into non-productive side reactions, so no wasteful byproducts are actually produced. Now the substances upon which an enzyme acts is known as substrates. The selective qualities of an enzyme are collectively known as specificity, and the specific site on the enzyme where substrate binds and catalysis occurs is called the active site of that particular enzyme. Okay. Now, regulation of an enzyme activity ensures that the rate of the metabolic reaction is appropriate to cellular requirements, that is essential to the integration and the regulation of the different functional activities. And it can be achieved by various ways. So enzymes can be used as an inhibitor, activator, hormonal activities, and also in the rate of synthesis or as synthetic enzymes. So specificity of an enzyme, how do you define the specificity of an enzyme? So it's highly specific compared to other catalysts. It catalyzes only a specific reaction. So it's like I was telling, the lock and key model, where it's very specific. So if you have a particular shape of the enzyme and it only substrates having that particular shape goes and binds over there, so it is absolutely fine because it's highly specific, because other shapes do not go and bind. But it's also about the catalytic ability. So specificity, enzyme can be highly specific but not highly catalytically efficient. But there are some enzymes that are not very specific but carries out catalytic efficiency with very high specificity. So it's just upon compromising upon specificity or catalytic efficiency. So there are three types of specificity: stereoselectivity, stereospecificity, optical specificity, reaction specificity, and the substrate specificity. So what is optical specificity? So it's the ability to recognize the optical isomers of the substrate, like it acts only on one isomer, like the D and L amino acid oxidase are actually very different. So there can be an enzyme that acts on only D and not L. So its ability to identify the D and L will help to increase the specificity. And isomers do not exhibit stereospecificity, because that it's the role of an isomerase to carry out the isomeric activity. So it will be not efficient for the isomerase to, if it's selective towards the stereo specificity, then it cannot convert from one isomer to the other. The reaction selectivity is the ability to catalyze only one specific reaction over the substrate. So it can undergo deamination, transamination, deoxycarboxylation, and each is catalyzed by separate enzymes. And the substrate specificity is it's specific towards the substrates, like glucokinase and galactokinase both transfer phosphoryl group from the ATP to different molecules. So there is absolute substrate specificity, relative substrate specificity, and broad substrate specificity. So absolute substrate specificity acts on only one substrate, that is urease. Relative substrate specificity acts on structurally related substrates. They further divide into group dependent and bond specificity. So group dependent relative substrate specificity acts on specific groups, like trypsin breaks the peptide bond between lysine and arginine. Chymotrypsin acts on aromatic amino acids. Bond specificity is specific to certain bonds, like the proteolytic enzyme glycosidase. They act on specific bonds. And broad specificity acts on closely related substrates, so hexokinase that can act on many hexoses. So the chemical nature and properties of enzymes. So the chemical nature is like protein or RNA. The tertiary structure and specific conformation essential for the catalytic power. You have the holoenzyme for different functional unit, apoenzyme and the coenzyme. So there are different chemical natures, each with a particular function. So they are allotted a particular function which they carry out. So each of them have different functions and different properties. So prosthetic group, so the difference between a prosthetic group and a coenzyme or a cofactor is like the prosthetic group is a non-protein molecule. It is tightly bound. It has a stable incorporation and cannot be dissociated. On the other hand, coenzyme or cofactor is the non-protein molecule. It's loosely bound, dissociable, separable by dialysis, etc. Yes. So yeah, that's all from the lecture part. Uh, if you have any questions, you can ask me. Maybe if you have any questions from the previous classes, that is also fine, because we need to also do that. Anyways, we will be having one 2 hour session after completing week 12 lecture. So we will be having 2 hours of class where we will be discussing a little bit of MCQs and some tricky questions, also some doubt clearing session. That is mostly, these are also doubt clearing, but that will be about from the live sessions. Anyways, if you have any questions, you can go ahead with asking me. I will be starting with the question and answers after some time. Like, I'm giving you a little bit of time to just ask me. If you have any doubts, you can just tell me now. Yeah, so no questions. Mm, no questions. Okay. Okay. So let us start with the questions then. The practice problems. Right. So this. Okay. So you people can give me an answer to this first, and then I will be discussing. So the site where the catalytic activity of an enzyme generally takes place is known as enzyme site, active site, activity site, reaction site. So the site where the catalytic activity of the enzyme takes place, what is it known as? Yes, that is correct. It is known as the active site. Okay. Active site is the correct answer. The next one, given the delta G naught for the reaction S in equilibrium with P is negative in the given direction S to P, reaction equilibrium favors the formation of which substance? P, S, both S and P equally, initially P and later S. So delta G naught over here is negative. It is saying in the direction of S to P. So the reaction equilibrium favors the formation of which particular substance? Yes, I have got one answer. Others, please try. So delta G naught negative is an indication that the reaction is feasible. So the reaction is feasible means the forward reaction is feasible. So S to P, so S is the reactant, P is the product. So P should, the formation of P should be more. So option A is correct, that is P. For the reaction S plus E forms ES, that is in equilibrium, to E plus P, where SEP stands for substrate, enzyme, product respectively. The quantities can be measured experimentally. So which of them can be measured experimentally? Substrate, product, total enzyme, bound substrate, bound product, bound enzyme, only substrate and product, or only free product. So which of them can you measure experimentally? Okay, so let us see. So what are the things that you're adding from outside? that is substrate, because you know the amount of substrate that you have added, so you can calculate that experimentally. You know the amount of product that is formed, so you can also calculate that depending on, you can do that experimentally by measuring what is the amount of product that is formed. You can do that. The other thing that also you can measure is the total enzyme, because you are adding the enzyme from outside. So if it's an, even if it's an in vitro, you can check it like about the amount of enzyme that has been produced, that is carrying out the entire activity. So the total enzyme, substrate and the product, all three can be measured. So option A is correct. Which of the following determines substrate specificity of an enzyme? Complementary geometry of the substrate and enzyme, complementary electrostatics of the geometry and enzyme, degree of hydrophobicity, degree of hydrophilicity of the substrate. So which of the following determines the substrate specificity of an enzyme? Complementary geometry of the substrate and enzyme, yes, that is correct. What else? Even the complementary electrostatics of the substrate and enzyme will also be correct. So A and B both are correct over here. But the degree of hydrophobicity or hydrophilicity will not matter, because it is not an interaction with the solvent but an interaction with another protein molecule. So there will be lot of other interactions like hydrophobic, hydrogen bond. So there will be a lot of interactions that are occurring. So A and B are the correct options over here. The next one, while monitoring the activity of the enzyme in UV spectrometer, which two parameters are monitored in the instrument? Absorbance versus wavelength, time versus wavelength, absorbance versus time, time versus absorbance. So check the x-axis and y-axis as well, because which will be in which axis, that you have to mention. So when you're measuring the activity of the enzyme, which two parameters do you monitor? Absorbance versus wavelength. Okay, anything else? Yes, option A is only the correct over here. Okay, a rate versus pH curve with inflection point at pH 4 suggests the involvement of or an dash in the catalytic step: hydrophobic amino acid, acidic amino acid, basic amino acid, or metal ion. So you're given, you get an inflection point at pH 4. So what does it indicate? So there is an involvement of what type of an amino acid? Hydrophobic, acidic, basic, or metal ion? So you have been given the pH of 4. C, B, no. So any amino acid that has an inflection point at pH 4 or in the acidic range, so there is an involvement of an acidic amino acid. So option B is correct over here. So if it had been, there is an inflection point at pH 9, 10, then it would have been basic. Okay. So choose the incorrect statement about a multi-step reaction. The free energy of the intermediate must be greater than the transition state. The transition state with highest free energy always corresponds to the rate determining step. Free energy of the intermediate may be greater than that of the reactant. By catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. So which of the following options are correct? Are incorrect? Is it 1, 2, 3, or 4? So you say 1, 2, and 3 are incorrect over here. Okay, let us see. The free energy of the intermediate must be greater than the transition state. The free energy of the intermediate must be greater than the transition state. This is incorrect, because from the intermediate step we go into the transition state. Okay, so intermediate is something like, so for example, you have a reactant and you're going to B. So in between A and B there is a transition state, and in between the transition state and A you have an intermediate state, and between the transition state and B you have another intermediate step. So the free energy of intermediate should be less, because the reaction is feasible towards the transition state for a reaction to occur. So one is incorrect. The next one is a correct statement where it says the transition state with highest free energy is the rate determining step. So that is correct. So if 2 is correct, then A, B, and C options are all incorrect. So it's D, because the last one, by catalyzing the reaction, the enzyme must decrease the free energy of the intermediate. That is also not correct. Uh, by catalyzing, because here you have to decrease the free energy of the transition state, not the intermediate. Okay, methionine synthase catalyzes the conversion of homocysteine into methionine in the presence of vitamin B12. Here, vitamin B12 plays the role of an apoenzyme, holoenzyme, cofactor, or prosthetic group. So, what is the role of vitamin B12 over here? So, there is a methionine synthase that catalyzes the conversion from a homocysteine to methionine. Yes, cofactor. So that is correct. So when B12 goes and binds to the methionine synthase, in the presence of that, the conversion occurs. So choose the incorrect statement about the enzyme. Compared to an uncatalyzed reaction, an enzyme speeds up the rate at which a chemical reaction approaches equilibrium. Enzymes make a reaction thermodynamically favorable. Enzyme decreases delta G reaction as well as delta G slash. And in the presence of an enzyme, all thermodynamic parameters of a reaction are altered. So which options are incorrect over here? So you're telling C, that is 1 and 4. Okay. So let us see option 4. In the presence of an enzyme, all thermodynamic parameters of a reaction are altered. This is not correct because there are only decreases the activation energy. Activation energy barrier. Okay. So only that parameter gets altered. The other parameters are not that much altered. So option 4 is incorrect over here. And the option 1 and 2, that is it speeds up the rate at which a chemical reaction approaches equilibrium, is correct, and enzymes make a reaction thermodynamically favorable, that is also correct. So option 3 and 4 is incorrect, that is option D will be the correct over here. That means enzyme decreases the delta G reaction as well as the G slash. The enzyme active site should preferably be more analogous to the product substrate transition state. So what should be the enzyme active site be more similar to? So it should look like what? Is it the product, substrate, or the transition state? So be it a lock and key model or any kind of a model, what should the enzyme look like? C, 1 and 3? No. Um, so it should more look like. So analogous means it should look similar to. So the active site of an enzyme should be more similar to the substrate because the active site binds the substrate, right? So option A is correct over here. That is 2 only. Okay. The next one is which group of enzymes would be involved in the reaction shown that involves the conversion of carbamoyl phosphate to carbamoyl aspartate. So which reaction or which enzyme should be involved? Is it the hydrolase, ligase, oxidoreductase, or the transferase? Yes, because there is a transfer of another group. Yes, so the transferase is involved. So you have the NH2 group over here that goes and attacks in the carbonyl carbon. This bond breaks, the phosphate is removed, and this particular group is added. So transferase. The next one is, so there is another chemical reaction. So which enzyme can carry out this particular chemical reaction? So there is some enzyme that is carrying out this reaction. So there is removal of ammonia and what else? Yeah, there is a double bond formation. No, ligase is not correct over here because it's not ligating anything. Even transferase is also not correct because there is no transfer that is occurring. And what about hydrolase? It should be either hydrolase or lyase because lyase does not use any kind of water molecules or something like that. So according to me, it should be lyase. Okay, so there is the removal of one hydrogen is moved, and CH4 methane is being, yeah, so it is lyase. Right. Okay. Yeah, that's all. Yeah. Anything else? Any questions or something? No questions? Okay then. Thank you very much for joining. We will meet again in the next week with a new lecture.